Hey guys, welcome to AP Biology, and we are going to be talking about our membrane transport, other forms of transport. This is lecture 4 in the series of the cell membrane and the structure and function of it, and this will be the last one. Okay, so as I say up here, the important part is to move it across that cell membrane. And there are multiple different types of transport, and some of the ones that we have not yet talked about are endocytosis. So endo, of course, means in, and cyto means cell, and osis or cis means a process of. So the process of bringing something into a cell is basically what endocytosis means. Phago means to eat, and pino means to drink. So bringing something into the cell to eat and bringing something into the cell to drink, not really quite to drink, but to bring liquid form along with smaller molecules is a example of pinocytosis. And then receptor mediated endocytosis, where first of all, something has to bind to a protein on the surface membrane. And this is the way in which many viruses get into our cells, in that they have a special protein that locks onto a receptor protein, and that receptor protein tells the cell, hey, this is something we need to bring into the cell. It's the virus's way of tricking the cell, so to speak. It is the key to get past that lock of the cell membrane. Okay, and one of the ways we can mess with viruses or prevent viruses from doing it is by creating things that either block that receptor protein or disable the key in some way. And exocytosis would be just the reverse of that, expelling large quantities of material out of the cell. Okay, so excuse me. All right, so let's look at this one, phagocytosis and pinocytosis. So here's a picture, this is phagocytosis. We see extracellular fluid, and we see this could be a bacterium. It could be a lot of different things. The cell is going to invaginate its cell membrane and basically create a food vacuole around that particle and then bring it into the cell, where oftentimes it will then fuse with things like lysosomes, which will play a role in digesting that down. And very similar process with pinocytosis, where it's taking in extracellular fluid along with small molecules in that. And we can see some pictures here. This is the pseudopodium, which is false foot of an amoeba engulfing a bacteria. And we can see how it's going around around that bacterium. And as you notice, that bacterium is almost as big as the amoeba itself. And over on the bottom transmission electron micrograph, we see the small invaginations of the cell membrane creating vacuoles, eventually creating vacuoles such as these over here, bringing particles into the cell. So once again, if you are at home watching this, I want you to click on the links here and watch these animations. Very important to watch both of these animations. They're only about a minute or two a piece, so it will not take you much time, but it will talk about some important concepts in those two short videos. So please watch those. And again, it's going to show you some great animations of these processes themselves. Okay, so let's look at some of the types of transport that we've already talked about. So look at the top diagram. Okay, and what do we see with the top diagram? We see particles moving straight through the cell membrane. Okay, and we see a low concentration on one side and a high concentration on the other, and the movement is going from high to low. So what type of transport is that? And if you guessed simple diffusion, awesome. What about the middle one? We see large molecules, such as a glucose molecule, going from high concentration to low concentration, but they're moving through a transport protein. What type of transport is that? And if you guess facilitated diffusion, awesome. And the bottom one, we see the little glucose molecules, but they're going from a low concentration to a high concentration, and we see ATP. And so hopefully you guessed active transport on that one. Again, so The main thing about active transport is that we're moving substances across a membrane against the concentration gradients. Again, it's like pushing a rock up a hill. This does require an input of energy, and it allows the cells to create basically concentration gradients, okay, where there's a higher concentration of something in the cell than there is outside of it. And this is a super important concept, and we'll talk about that. Okay. So, again, concentration gradients equal work, because once you have a concentration gradient, it's like water behind a dam. We can now use that water behind the dam, run it through a turbine, and generate electricity with it. Okay, the same thing with a concentration gradient. Once you have a high concentration of a substance on one side of the cell membrane, you can use that high concentration gradient to do other types of work, whether it's transporting something across a cell membrane, or it's being used to generate energy in the form of ATP, which we'll get into when we talk about cellular energetics. Okay. One of the most important transport proteins that is involved in creating these concentration gradients is the sodium-potassium pump. Okay, and as I say up here, this sodium-potassium pump, which can be found in a variety of cell types, specifically in neurons and also in cells that line your intestine. Okay, we find lots of these sodium potassium pumps, but they're basically found in all cell types. As you can see, about 25% of your daily energy expenditure goes just to powering these pumps. And you did the models of this in class the other day, and I just want to kind of go over it a little bit. And so, what does the sodium potassium pump do? As you can see in this intracellular space, we have a low concentration of sodium ions and a high concentration of potassium ions. And so the sodium potassium pump, when it is open to the intracellular side, the sodium binding sites are in such a way that they can accept sodium ions but cannot accept potassium ions. As you see, there's no binding spot for potassium ions here. So sodium ions are going to bind to the sodium ion site. And as you can see, there's enough for 3 sodium ions to bind there. Okay, so 3 sodium ions will bind. That's going to cause a shift in the protein when ATP binds to it. And when ATP binds to that protein, it's going to phosphorylate it. What does that mean? It means it's going to donate a phosphate group. The donation of that phosphate group is going to cause a shape change in that protein, which then causes the sodium ions to be released into the extracellular space. And now the binding sites for the potassium are going to become receptive to potassium ions, and the sodium binding sites are going to become non-receptive to sodium. So now sodium ions, which are at a low concentration outside the cell, will bind to this pump, and the protein is going to dephosphorylate. What does that mean? That means the phosphate's going to fall off. And when that phosphate falls off, the protein reverts back to its original shape, as we see over here, and moves that potassium against its concentration gradient. That's how the sodium potassium pump works. Okay? And we'll see a couple of different examples of where we use that. Oops. Okay. Just trying to. All right, so here's again a picture of the sodium potassium pump walking our way through it. We see the 3 sodium ions binding. Over here, we see phosphorylation of ATP. That's going to cause a protein shape change. And as you can see, we see a change in the shape of the binding sites of both the sodium and the potassium. Potassium now can bind to this changed shape protein. Notice that the phosphate group is still on there. When that phosphate group falls off, the protein reverts back to its original shape and then transports the potassium into the cell. Good one to know, and to know how active transport works. Okay, so where do we use that? Well, most importantly, inside of our nerve cells, in our neurons. Okay, here we see a picture of a neuron. and we see a lot of sodium outside and we see a lot of potassium inside the cell. Well, there are potassium transporters and there are sodium transporters that allow for the movement of both sodium and potassium. And these actually have little gates on them. They open and close uh, as a result of stimulation. Okay. And so basically, what's going to happen is that the sodium gates are going to open and sodium's going to come rushing inside the cell. And that's going to cause a depolarization. What does that mean? Well, if you notice that the cell is negative on the inside and positive on the inside, on the outside. And basically, what's going to happen when all these positive sodium ions go rushing in is that the inside of the cell is going to become briefly positive in the region of where that's rushing in. And that's going to cause a voltage change. And that's basically what a nerve impulse is. And then those gates close, and then the concentration gradient is reestablished once again by the sodium potassium pump. It's a little bit more complicated than that. We'll get into that when we talk about cell communication a little bit later in Unit 5 or 4. Okay? And one of the ways I always like my students to remember it, it's a salty banana. We got lots of potassium, which is what bananas have inside the nerve cell. And if you have salt on the outside, and that gives you the setup of a neuron, okay? Salty banana, remember it. Lots of potassium on the inside and lots of sodium on the outside is the normal conditions. And again, like I said, that establishes a voltage difference between the inside of the cell and the outside of the cell. Well, you may ask, well, potassium ions are positive and so are sodium, so why is that difference? Well, the difference inside the cell is because there's a lot of other negatively charged ions and proteins and DNA inside that cell, which is what causes that cell to be relatively negative to the outside. It's about a minus 70 millivolt difference, okay? But when those sodium gates open up, it's that voltage difference is going to reverse itself, okay? And that's what's going to happen. And again, we'll talk about that when we get into Unit 4. All right, another type of active transport pump is called a proton pump. And proton pumps, just like their name indicates, pump protons, which are hydrogen ions, from areas of low concentration to areas of high concentrations. And once again, we set up this concentration gradient, and we're going to see this specifically when we talk about how ATP is made in the mitochondria. But proton pumps are also used to move things like sucrose in plants into the phloem cells. What's a phloem cell? Phloem cells are transport tubes that transport sugars from areas where those sugars are produced to areas where they are stored. So where they're produced is called the source, and where they're stored is called the sink. And it's generally going against a concentration gradient. So first of all, we are going to move sucrose from an area of low concentration to an area of high concentration. So you would think that's active transport, but there's no energy being shown here as there is here. So how does that happen? Well, we use active transport to establish a proton gradient. And then we use the facilitated diffusion of the protons that are going to pull the sucrose molecules with it down the concentration gradient. So the diffusion of the protons is so strong, the process, that it is able to pull a sucrose molecule against its concentration gradient across. So this is a type of facilitated transport that is only made possible due to the active transport of these protons to establish that concentration gradient in the first place, and then using that concentration gradient to move sucrose from an area of low concentration to an area of high concentration. Okay? We're going to see something very similar in the blood cells also, or kidney cells, or the cells that line your small intestine. I'm going to go over this diagram a little bit in some better detail in a couple of slides here, but I just want to show you is that we have a sodium glucose transporter here. This is our co-transport molecule. You have a glucose transporter over here, and we have a sodium potassium pump. And all I want to say right now is that we're going to establish a concentration gradient, use that concentration gradient to move glucose into the cell, and then glucose will leave the cell via facilitated diffusion. And the organ that really utilizes this all the time in terms of active transport is your kidneys. Okay. Now I do want to set this up a little bit. Up here we see the cortex of the kidney. What does that mean? We're above this dotted line. That is the outside edges of the kidney, and below we see the medulla. And what I want to say right here, from the top of the cortex down to the bottom of the medulla, the concentration of salts and mostly of sodium chloride increases exponentially. So we have very low sodium chloride concentration up here and very high sodium chloride concentrations down here. So we can see all sorts of transport going on. We see water and some bicarbonate ions and things that are leaving via simple diffusion. And then down here inside the kidney, where it starts getting very more concentrated in terms of salts, we see water and salt leaving via simple diffusion, urea and water also leaving via simple diffusion. But then we also see active transport, which is the red arrows, the passive transport with the blue arrows. We see some salt being actively pumped out in the less concentrated regions of the kidney, i.e. in the cortex. So it's actively being pumped out. Up in here, also glucose is being pumped out of the cell, and we see that all happening. And we also see some active transport down in this region. So the kidney does a lot of both active and passive transport. And here's a graph that shows you a little bit of how efficient your kidney is. Your kidneys filter about 180 liters of water per day. So that means that's how much water is being pulled out of your kidneys. But you don't pee 180 liters, you only pee about 1 liter. So the kidneys reabsorb 179 liters of that. Look down at chlorine. Okay, the kidneys filter 630 grams of chlorine, but you only pee out 5 grams of chlorine. So that means that your kidneys are reabsorbing most of that, and the same with sodium. And look at glucose. Okay, glucose, your kidneys filter out 180 grams of chlorine, and it reabsorbs almost all of that. If you have type 1 diabetes, which stands, the actual scientific name for diabetes is diabetes mellitus, which basically stands for honey urine. Okay, because back in Roman times, when this condition was first diagnosed, they diagnosed it by tasting the urine, and if it was sweet, that was an indicator that the child had diabetes. And most children up until about the 1920s generally died of type 1 diabetes by 5 or 6 years old, because they were peeing out all of their potential energy, i.e. glucose, and it wasn't being used in their bodies, and their bodies were basically consumed from inside out. Okay? And again, we'll get into some more detail of that a little bit later. So let's look at this particular diagram, and this is an important diagram to know. What you see here is this is the glomerular capsule. This is where the blood is being filtered, and this would be the filtrate. And you can see that there's a high level of glucose in here and a high level of sodium. Okay, and so, or I should say, we have a high level of glucose, and we also have some sodium in here. Now, this is the sodium glucose transporter. So what's going to happen here is that the sodium is going to diffuse down its concentration gradient. So this is a relatively high concentration of sodium, as we saw in that earlier diagram. Look up, we go back and we'll see that there's a lot of sodium in that filtrate and also some glucose. The sodium is going to filter down its concentration gradient. There's a relatively low level of sodium inside of this particular cell. And then so sodium will pull the glucose with it. Why is there a low level of sodium inside the cell? We have all the sodium coming in. It's because we have a sodium potassium pump down here that is moving sodium from a relatively low concentration in here out into a higher concentration near the blood. So it's moving it out, okay? And we're moving sodium, we're moving potassium into this particular cell. So this helps to maintain a low concentration of sodium within the cell, a higher concentration outside. So sodium is going to diffuse down through its transport, its concentration gradient through this co-transporter, pulling the glucose with it. And glucose is relatively high now in concentration within this particular cell. And the other glucose transporter protein acts as basically facilitated diffusion because we have a high concentration of glucose inside the cell and relatively low concentration out here. So the glucose will now diffuse down its gradient into the blood capillary. And that's basically how we get glucose into our blood and into our various cells, because that blood will now carry that glucose to different parts of the body where it is needed. So that's a good diagram to know and understand how it works. Okay. Endosymbiosis, again, kind of a version of bulk transport, and shows how we know from the data that we have and the information that we have how eukaryotic cells basically evolve. And once again, eukaryotic cells their origin is ancient prokaryotic cells somewhere about 2 billion years ago, where different organelles were brought into the cell. Actually, not organelles, different bacterium were brought into the cells. Okay. And again, in this particular case, what we have are the fusion of the bringing in of two different bacterium here, one that produced energy, and here is its membrane and its DNA down here. And it's brought in through phagocytosis. And eventually, what we see is the incorporation of these. Maybe this was a heterotrophic bacterium that produced ATP, or maybe it was a photosynthetic bacterium that was able to take sunlight and make sugars. In both cases, the process was the same: endocytosis, and eventually the creation of a modern eukaryotic cell. And the evidence that supports that, both chloroplasts and mitochondria have a double membrane. Okay, that's a result of that phagocytosis. They have their own DNA that's in the same form as we see in a bacterium. It's a single circular chromosome. Their ribosomes are almost identical to bacterial ribosomes, which are different than eukaryotic ribosomes. The other thing we see also is that they have certain proteins that are only found within in bacteria, are also found in chloroplasts and mitochondria. And lastly, both chloroplasts and mitochondria are roughly the same size as a bacterium. So those are all pieces of evidence we have for endosymbiosis. And we're going to read this article in class next week. So we'll do a little exercise with that particular article. So just looking at endosymbiosis version 2. Alrighty, that's it for this particular podcast.